Viral replication is where you start to see what a virus really is. Beyond the shape of the virion, you look at what they really are. But also, it will all start making more sense, and the purpose of the different things will start making more sense. You have to work through some biology stuff to understand viral replication, but once you work through it and understand it, it will fall into place and you will know what a virus is. And most people don't. So let's get into it. So viral replication is critical or absolutely necessary for any virus to keep going. It has to replicate in a host cell. So we call them obligate intracellular parasites. That means they must grow inside a cell. There are also bacteria that are obligate intracellular parasites. Viruses are crazy in that way. They don't grow. They don't get bigger. They are assembled at full size. So the cell is kind of like a machine that builds the virion. So the virion does not change size. It just gets made at its size. And that's inside of a host cell. And that process of replication is going to depend on what the host cell is. So we will look at a bacteriophage first, because that's the simplest situation, and then we'll look at animal viruses. And you'll see there are some steps that are different between them because of the host, how different the hosts are. And the big thing is that animal cells do endocytosis. So animal viruses use endocytosis to get into cells. Bacteria and archaea don't have vesicles. They don't use endocytosis or exocytosis because they are smaller than a vesicle. And because they have cell walls, so it wouldn't make any sense anyway. So bacteriophage can't use endocytosis to get into a cell. So if we look at bacteriophage first, they have to go through 5 steps. The first one is attachment or absorption. They are the same thing. It's just chemically we think of it as absorbing. Biochemically we think of it as attaching. It's the same process. And so where does that happen? Kind of like here. This is the phage. This is the cell. The phage attaches. And then penetration is when it injects its nucleic acid into the cell. So it has to enzymatically digest the cell wall and then enzymatically push the nucleic acid into the cytoplasm. Then somehow this nucleic acid has to have instructions on it that this cell will follow to make the pieces of new virions, to make new nucleic acids, to make the capsid and capsomeres, to make anything else the virion would have in it. And then maturation or assembly, these are the same thing. It's the process of fully assembling the virions. And this all happens in the cytoplasm in a bacterial virus. And then the last thing is release. This is where a bacteriophage is typically going to lyse the cell and get out that way. So this cell dies during this process. So in a little more detail, let's look at how a bacteriophage replicates. So how does a virus that infects a bacterial cell, how does it replicate? And we're going to look at this kind of a little bit more step by step. So the first step is attachment, and this is really a critical step. This is really the make or break step for most viruses. If the cell is going to prevent a virus from replicating, the cell usually has to prevent this first step of attachment. So what's typically going to happen is these proteins on this bacteriophage virion, these tail proteins, they will attach to different proteins in the cell membrane. So I know this looks kind of like a bizarre looking insect or spider or something, but really what you're seeing is proteins whose job it is to find and attach to specific proteins in the cell membrane. And those are going to anchor this whole thing in place. And that's going to set up the next step, penetration. And so in the next step, somehow this has to break the peptidoglycan layer. And so it has enzymes that can digest the peptidoglycan layer. And then it's going to inject its nucleic acid into the cell cytoplasm. So the nucleic acid was stored in the capsid, or in this head. And then once this virus has breached the peptidoglycan layer, it will inject the nucleic acid. And for bacteriophage, the best known examples are DNA viruses, but there are RNA viruses also. So let's keep going. Biosynthesis is going to be, in a lot of cases, the most complicated step, or the step that you're going to have to work to understand. That's because this is the process of creating all the new materials that will go into the new virion. So if the host cell becomes a virus factory, what it's going to do is make new virions, and biosynthesis is where that making happens. It's where the raw materials are made. So the nucleic acids that have to get made, the proteins that have to get made. And so you're going to see later in a detailed example all of that. Well, if it's a DNA virus, like what we're looking at, it has to transcribe the genes and then translate them to make proteins. What do those proteins do? Well, the first proteins are going to change the cell's behavior. The virus is going to change the way the cell acts. And specifically, typically they take over protein production. So they're going to kind of hijack that system. Some viruses are going to break down the host DNA, and that will ultimately kill the cell. Some will disrupt host transcription. Some will disrupt host translation, as all this stuff we can understand. Maturation and assembly and release are all pretty straightforward. The more detail you look in, the more you try to understand these at the biochemical level, the more complex they get. These are all incredibly complicated, but we can understand what's happening without going into that level. Let's see, let's keep going. So then we look at animal viruses and how they replicate. And again, they're different because they get in a different way. They can use endocytosis or membrane fusion. And that means once they get into the cell, what gets in is the capsid, and it has to be broken open. So that means the animal virus has to uncoat. And then eukaryotic cells are just different. They're more complicated. So in a eukaryotic cell, there's a nucleus. That's where the DNA is. That's where transcription happens. And then messenger RNAs are sent into the cytoplasm. That's where translation happens. Some translation happens, well, the proteins get made essentially in the lumen of the rough endoplasmic reticulum, and then they get sent through vesicles to the Golgi, and they get sent through vesicles all over the cell. Bacteria don't have anything like that. And so animal viruses have to take control of all of those systems, or use all of those systems. And that's what I'm talking about with maturation and release. Animal viruses will tend to use something similar to exocytosis or membrane fusion to get out of a cell. Okay, animal viruses have the same general goal as what bacteriophage have. They want to make more virions, but they have to take control of a different kind of cell to do it. And so that means they have to add a completely new step. They have to add an uncoating step. So again, the first step in replication of an animal virus is going to be attachment or absorption. Absorption is kind of the word we use when we talk about a small thing sticking to a larger thing, and that really is what it's like when a virion sticks to a host cell. But they mean the same thing in this case. Again, it's the critical step where something has to match perfectly. That is, the virus has to perfectly match certain receptors on the host cell surface, or the other way around. There has to be that perfect lock between a receptor and what it detects. And usually the virus is going to mimic what a host receptor is looking for. And so it can't be overstated how important this step is, because this is what determines which kinds of cells a virus can infect. So one clear example is the SARS-CoV-2 virus that causes COVID-19. It can infect any cell in the body that expresses the ACE2 protein on its surface. And so if we can look at which cells in our bodies have that protein, that really does predict what parts of the body that virus is going to affect. And age-related trends in where that receptor appears in the body determine some of the age-related differences in COVID-19. And we will see that in more detail when we look at COVID-19, but it really is important. And if the body can change the receptors in some way, the virus can't attach. And so really the way we think about it is, if a virus successfully attaches to the host cell, it's pretty much over for the host cell. So again, this is the first step. And then the next step has to be penetration, and somehow the virus is going to go into the cell. This is going to be different from what we saw in a bacteriophage, and that's because animal cells don't have cell walls, and that means they can use vesicular transport. They can do endocytosis and exocytosis, and viruses use that. Animal viruses can use that when they try to get into an animal cell, whereas a bacteriophage doesn't have that option. So there are two kind of ways this looks in an animal virus. One is receptor-mediated endocytosis, and the other is membrane fusion. Either way, it starts with the spike proteins attaching to receptors on the surface of the host cell. And so in the next slide, I'm going to show you kind of two different ways that can look, and those two different processes. So this is the two sort of situations we get in different viruses, where what we've done is draw the cell membrane like this, or we've drawn the cell membrane like this. And what we're showing is two different ways animal viruses will get into cells. This is going to be endocytosis, or receptor-mediated endocytosis. So the spike proteins on this virion are going to attach to various receptors on the cell surface. Ultimately, that is going to form kind of a sphere around them, and that will pinch off when this touches this. And then this virus will be inside a vesicle in the cytoplasm. This virion will be inside a vesicle. The virion has to have a way of getting out of the vesicle. So it has to have a way of breaking that open. And then it has to have a way of getting the nucleic acids out of the capsid. So there has to be some way of breaking this open also. If they use membrane fusion, this makes use of the envelope. Remember, there's a lipid bilayer in enveloped viruses. And so with membrane fusion, what happens is the virion gets really close to the cell membrane by attaching to lots of other molecules on the cell membrane, and ultimately the membranes fuse. And so then what would happen is this would become cytoplasm. This would become inside the cell. Imagine two soap bubbles touching each other and becoming one soap bubble. That's what this is like. And so what that does is that leaves the envelope in the cell membrane. So this envelope fuses with the cell membrane and it stays behind here. And the capsid ends up in the cytoplasm. And the capsid has to somehow release the nucleic acid. So a lot of viruses do both of these. Whichever one happens first seems to be what they do. So I know influenza can do both. SARS-CoV-2 can do both, and those are the two viruses we're going to look at in the most detail. Uncoating, we won't get into how it works, but again, that's getting the nucleic acid out of the capsid. Biosynthesis is going to be tricky. This is where the capsid, or the capsomeres, have to be made. Those are the capsid proteins. Any viral enzymes that it's going to need have to get made. Spike proteins have to get made. New nucleic acids have to be made, so it has to copy the genome so that that can be put into new, virions, new virions. This is the step where the genome type matters. So if it's RNA or DNA, if it's positive sense RNA or negative sense single-stranded RNA, that is going to dictate what happens during biosynthesis. These jobs all have to get done, but how that works depends on what kind of genome they have. Maturation and assembly, you'll see they have to take over a lot of parts of the cell, like the endomembrane complex, the endoplasmic reticulum, the Golgi, the vesicles. Oops. Okay, and I'll show you this in some diagrams, and it will make more sense. When we go through in detail and we look at how influenza replicates, you'll see these in action. There will be a few extra things happening because any real virus is more complicated than this, but you will also, once you work through it, you'll understand it. And then the last thing is release, and that's either breaking open the cell and killing it, or leaving through a process like exocytosis. And in cases where the virus leaves through exocytosis, it's not like the cell is happy. The host cell is now a factory for making these virus particles. So the cell is now a zombie that just makes virions. So this is a diagram from your textbook that shows the different steps, and this would be an exercise you could do. What they have in the textbook is little numbers up here, and the numbers for each of these steps correspond to numbers here. So, I don't know, see if you can figure out which number would be which. It's not meant to be challenging here. It's just a thing you could do. And it's always good to look at different diagrams of the same thing to see what do the artists show the same way, what do they show in different ways, and why do they choose to draw certain things. That's enough for this video. In the next video, we're going to look at biosynthesis and how it differs between different types of viruses, and that will be its own thing. We need to take some time with that. So I'll see you there.